I'd like to welcome you to the pattern formation session. I will be co-chairing with Anna Bustoria and our postdoc co-chair, Mo Huang from Eric Wieshaus's lab. And our first speaker is Bonnie Weasner, and the title of her talk is Sulfate Transformations in Sine-Oculis Eye-Specific Loss-of-Function Mutants Obscures Direct Regulatory Interactions Within the Retinal Determination Network. Did I get it right?
You did. It's a rather long title. Sorry. So thank you. So I'd first like to thank the organizers for giving me the opportunity to present my data, and all of you for coming to listen. The story I'm going to focus on today is actually a really small part of a much larger project, but we feel that it serves as something of a cautionary tale regarding the importance of looking at the overall state of a cell or a tissue, particularly when attempting to determine regulatory interactions, simply by looking at loss-of-function expression and loss-of-function mutants. In the Kumar lab, we study the early specification and differentiation of the Drosophila Compound Eye, which is seen here in this micrograph. The compound eye is composed of 800 individual units, or ommatidia. Each ommatidia is composed of approximately 20 cell types, including photoreceptors, which are the trapezoid structure that you see here in this serial section, lens-secreting cone cells, bristle cells, and pigment cells. The adult eye is derived from the Eye-Antennal Imaginal disc, which is a monolayer epithelium, and is outlined here in red with the eye portion on the left and the antenna on the right. Proliferation, growth, and segregation of the eye and antennal portions of the disc will occur during the first and second larval instar. During third instar, photoreceptor differentiation will begin with the initiation of the morphogenetic furrow at the far posterior edge of the disc, and it will progress anteriorly as development proceeds. And the furrow marks a physical separation between the undifferentiating proliferating cells ahead of it and the ordered differentiated photoreceptors behind it, which are seen here in green. And this process of retinal specification is overseen by what is commonly referred to as the Retinal Determination Network. And to date, this network is composed of 14 transcription factors that were initially identified by loss-of-function phenotypes that result either in a disruption or a complete absence of the adult eye, as seen here, as well as their ability to induce eye development in non-retinal tissues. However, unlike the name would suggest, the eye-antennal disc does not just give rise to the eye and antenna, but rather multiple adult structures, including the ocelli, nearly all of the surrounding head capsule and bristles, and the maxillary palp. And so because all of these adult structures initially arise from the same set of cells, one very broad question that our lab is interested in addressing is how regional sulfate specification is established and maintained in this disc. And as part of that endeavor, our lab, as well as others, have recently begun working on a new model or a new way of thinking about the retinal determination network, in that in addition to promoting the primary eye fate, it is actively repressing secondary fates, and that it's this juxtaposition or sort of battle of regulatory networks, if you will, that's ultimately responsible for the regional specification of the disc. And this is a model that we think applies broadly to other GRNs and other tissues. So understanding the regulatory interactions between members of a GRN as well as with members of other GRNs would be critical for understanding how this process of promoting a primary fate and repressing a secondary fate is actually achieved. So much of the work in our lab focuses on refining the regulatory relationships that have been observed for the RD network. And so with that in mind, for my talk, I'm going to focus on the very specific question of whether the retinal determination gene Sine-Oculis regulates eyes absent during retinal development. So a brief background on Sine-Oculis, or eyes absent, or SO and I as I'll refer to them. Sine-Oculis is a highly conserved member of the 6 family of proteins. It contains a homeodomain and functions as a DNA binding transcription factor. Eyes absent is a highly conserved member of the Ia family of proteins, and they function as transcriptional coactors and protein phosphatases. Both of these proteins are absolutely critical for eye development, as loss-of-function mutations in either gene will result in the complete ablation of the adult eye. Furthermore, during retinal development, SO and IA form a biochemical complex, and this complex functions as a bipartite transcription factor, with SO binding DNA and I's absent functioning as a transcriptional activator. Its phosphatase activity has been recently determined to be dispensable for eye development. And so this complex will go on to activate important downstream targets that are necessary for eye development. It has been suggested for some time that SO and IA might regulate each other's expression, and this was based on the observation that in loss-of-function mutants, there's a corresponding loss of the partner protein. And by that, what I mean is if we look here in an I's absent mutant that's been stained for Sine-Oculis protein, you can see that Sine-Oculis is missing in the majority of the remaining eye disc. And the reciprocal relationship can be seen in a Sine-Oculis loss-of-function mutant that's been stained for I's absent. There is some expression that you do see here in the dorsal part of the disc. That's the region that gives rise to the ocelli, and those are specified independently of the retina. So we've taken this analysis, this observation a step further in collaboration with Arash Basharulah's lab, and we conducted QRT-PCR experiments on late third instar, IA2, and SO1 discs. And what we have here are the relative transcript levels of Sine-Oculis and I's absent in both mutants. And what you can see is that the transcripts are significantly reduced. And so we thought, taken together, that these observations provided a strong foundation for this idea that Sine-Oculis might be regulating I's absence expression. Furthermore, if we examine the I's absent locus, we see that there are multiple predicted SO binding sites, as well as ChlP-Seq peaks that were recently identified by Graham Marden's lab. And although previous work in Nancy Bonini's lab had identified an eye-specific enhancer element that contains a Sine-Oculis binding site, and when deleted, results in a complete absence of the eye, we were curious if there actually were any other enhancer elements within this locus that are actually being masked in this mutant, and whether or not those were actually responsive to Sine-Oculis. So through a lot of data that I don't have time to discuss today, we conducted an enhancer bashing screen of the eye locus, and I determined that a composite enhancer that is composed of 3 distinct cis-regulatory elements is responsible for all temporal and spatial expression of I's absent in the retina. And each one of these individual enhancer elements is activated at a specific time and location in the disc, but only when they function together can you fully recapitulate I's absent expression. So what I have here is a time course of I's absent expression through larval eye development, and what I hope that you can see is that the enhancer LAC-Z reporter corresponds quite beautifully to the endogenous protein. I took this a step further by conducting loss-of-function rescue assays in which I fused the individual enhancer elements as well as the composite enhancer elements to an I's absent cDNA and assayed their ability to restore eye development to loss-of-function mutants. And only the composite enhancer was capable of fully rescuing the no eye phenotype. So since we're ultimately interested in whether or not Sine-Oculis was regulating I's absent, I next decided to see whether or not this enhancer element was activated in Sine-Oculis mutants. And it was our expectation, based on the QRT-PCR data and the expression data, that this element should be off in a Sine-Oculis mutant. But we were quite surprised to see that the enhancer was still strongly activated both in Sine-Oculis SO1 mutants as well as in SO3 null clones. But more importantly, what we saw is that in the clones, I's absent protein was still present, suggesting that the phenomenon we were seeing was not simply due to the perdurance of LAC-Z. And this was quite perplexing given this earlier data that we had. So now the question for me sort of became, why are we losing I's absent expression in a Sine-Oculis mutant? And one hint that I got from what might be happening was from an earlier paper from Walter Gehring's lab, in which they showed that I's absent was still present in discs of late second or early third instar Sine-Oculis mutants. So this made me think that, well, maybe what we're seeing is actually a progressive loss of I's absent over time. So I decided to re-examine I's absent expression in these mutants over developmental time. And indeed, what we see is that that's exactly what is happening. So at 72 hours after egg lay, which is the beginning of third larval instar, 100% of the discs examined still had strong I's absent expression. But as development proceeds, you can see that the expression becomes weaker until at 168 hours, the majority of the discs show no I's absent expression in the disc at all. So now I sort of wanted to know, well, what would be the potential causes for this loss of eyes absent? And the simplest explanation was that the retinal progenitor cells are undergoing increased cell death. And this was based on the fact that previous studies have shown that Sine-Oculis mutants have high levels of cell death in the retinal progenitors. And we had a few years ago shown that in eyes absent mutants, a similar phenomenon happens, but we have a wave of cell death that begins at the beginning of third instar and it continues. But then at approximately 108 hours after egg lay, this cell death precipitously drops off, and we found that there was a subpopulation of retinal progenitors that were still present, and we identified those by different markers for retinal progenitors. But even more surprisingly, we found that these progenitors were now simultaneously expressing non-ocular genes, for example, CUT and orthodentical, which specify bristle and head capsule respectively. We found that CUT and orthodentical had actually expanded to cover the entire portion of the remaining eye disc, suggesting a gradual transformation to head capsule. And this gradual transformation corresponded quite nicely, the de-repression of these genes corresponds quite nicely with the wave of cell death. So given the phenomenon that I saw in eyes absent mutants, I was curious if the same thing was happening in Sine-Oculis mutants. And indeed, when we look at the expression of CUT and orthodentical, we see that at 96 hours after egg lay, which was the first time point in which we see a reduction in eyes absent expression, we saw that CUT, which is not normally expressed in the disc, had expanded into the eye disc. At 120 hours, which is the first time point in which I would find any discs that had no eyes absent expression at all, orthodentical and CUT had both expanded. And then finally at 168 hours, which is incidentally the time point at which we did our QRT-PCR analysis and our expression analysis, CUT and orthodentical have expanded to cover the entire eye disc. So we're left with a model that as eyes absent expression decreases, CUT and orthodentical are being de-repressed, suggesting a gradual transformation to head capsule. And furthermore, when we look at SO3 clones, we see often that these null clones that are in the middle or the margins of the disc have actually have no eyes absent expression. When we look at the adults, we actually see that there is a head capsule that corresponds to these regions. So we think that our interpretation is those clones which are lacking eyes absent expression have undergone a transformation to head capsule. So just very quickly to summarize, the take-home message from this is that cell death and cell fate changes in Sine-Oculis loss-of-function mutants are obscuring our interpretation of the proposed regulatory relationship between these two genes. And in support of this conclusion, I've shown that the main cis-regulatory elements responsible for eyes absent expression are still expressed in Sine-Oculis loss-of-function mutants, but more importantly, eyes absent protein is still expressed in these clones, suggesting that SO is not necessary for the initiation of eyes absent, and that instead the protein is being lost progressively over time through cell death and a gradual transformation to head epidermis. So I just quickly want to acknowledge some people, all of the members of the Kumar lab, particularly Brandon Weasner, who did a lot of the cloning for the enhancer bashing project, Arash and Sarah, who did the QRT-PCR experiments, these individuals for reagents, and the Eye Institute for funding. And just quickly, I want to make a plug for my fellow lab mates that are presenting posters here. Each have a very unique and interesting story that all relates back to that general question of how regional cell fate specification is achieved in the disc. And if that's a question you're interested in, please go see them, because they would be very happy to talk about that. And with that, I'd just like to thank you for your attention, and I'm happy to take questions.